So we took two molecules of pyruvate and we went through the pyruvate dehydrogenase. Now we have two molecules of acetyl-CoA. Each acetyl-CoA is going to go through a cycle called the Krebs cycle. The Krebs cycle is a cycle that occurs inside the mitochondria, inside the matrix. So inside this area of the mitochondria. And it is going to yield more NAD, and this time also a molecule called FAD that we talked about a few lessons ago, which is going to basically yield more electron carriers. We are also going to see that we have different products as well. So this is a simplification of the Krebs cycle because we don't actually need to know for the IMAT and BMAT exam the names of the enzymes and steps. However, the following illustration actually shows you exactly the highest yield points you need to know for the IMAT and BMAT exam. So acetyl-CoA is going to be incorporated together with another molecule from the end of the cycle, and it is going to create a 6-carbon product. This carbon product is going to go through decarboxylation, and we are going to get our first product, which is carbon dioxide, which is a waste product. Then the next are going to go through the dehydrogenation, and we are going to charge NAD molecule. Then we are going to do it once again to the next. So we are going to have another NADH molecule. Then we are going to do another decarboxylation. So far through this metabolic pathway we have, for one acetyl-CoA molecule, for one pyruvate, we are going to have 2 carbon dioxide and 2 NADH so far. Then through this process, this step, we are going to yield ATP. This is the only ATP we are going to yield from one pyruvate, one acetyl-CoA molecule. So each pyruvate, each acetyl-CoA is going to give us 1 ATP. Then we are going to continue. And this time we are not going to get NADH, we are going to get FADH2. So we are also getting FADH2. Now one last thing would be to get from another step another NADH. So in total we are going to have 3 NADH molecules, 2 carbon dioxide molecules, and 1 FADH2 molecule for each acetyl-CoA. So we are going to have in a single cycle 2 carbon dioxide, 1 ATP, 1 FADH2, and 3 NADH. Now, because again, which is so important, because we are going to do and have 2 cycles for 1 glucose molecule, we are going to have 4 carbon dioxide, 2 ATPs, 2 FADH, and 6 NADH, which is so important to remember. So if you see on the IMAT and BMAT exam a question that asks you about 1 acetyl-CoA production or 2 the products of glucose metabolism or the acetyl-CoA metabolism, you need to remember what are the products of the glycolysis, link reaction, and Krebs cycle. Now that we have the Krebs cycle and we have so many FAD and NADH that are available to actually carry the electrons to somewhere else, we are going to learn about a special place inside the mitochondria, which is going to be called the electron transport chain. And the electron transport chain is a sequence of molecules, a sequence of structure, which is basically going to act as some sort of a chain, obviously, which is going to deliver between the different molecules the different electrons that the coenzymes, the electron carriers, actually carry. So they are going to come. This is the NAD or FAD. They are going to basically take the electrons that they are carrying on top of them, and they are going to put them on these structures on the inner membrane of the mitochondria, which is called the electron transport chain. When the electrons are going to drive and to jump between the different structures in this electron transport chain, we are going to see how we can actually yield energy when they move through the different structures in this electron transport chain. But this is a topic for the next lesson. I will see you next time.